Hi viewers, welcome to my classroom. Today we can discuss about transcription. The term transcription means the process of transfer of genetic information from DNA to RNA is called a transcription. DNA makes RNA, RNA makes protein. That is called central dogma process. Here the process begins. Transcription factors assemble at a specific promoter region along the DNA. The length of DNA following the promoter is a gene and it contains the recipe for a protein. A mediator protein complex arrives and carries the enzyme RNA polymerase. It steers the RNA polymerase, setting it with the type of other factors initiating the process of transcription. Transcription factors bind to specific region of DNA which will unzip or which will unwind the DNA double helix and through which the RNA will be transcribed or which will be produced through the groove. We can see in the schematic diagram and explanation. Examine the structure of a transcription unit. The reference is made with respect to the polarity of coding strand. The promoter is located towards 5' end of the structural gene, and the presence of a promoter in a transcription unit that also defines the template and coding strand. By switching its position with terminator, the definition of coding and template strand could be reversed. The terminator is located towards 3' end, that is the downstream of the coding strand, and it usually defines the end of the process of transcription. There are additional regulatory sequences that may be present further upstream or downstream to the promoter. From the previous slides, I think you could understand what are the major unit of transcription. So in the next slide, we can see how the process of transcription take place in prokaryotes or in bacteria. Here you can see 3 steps in transcription: initiation, elongation and termination. See, in these 3 processes, actually what is happening? RNA polymerase and sigma factor bind to the double helical DNA, then the production of RNA take place, and finally separation of RNA polymerase and DNA is taking place. Let us see what is happening during prokaryotic transcription in the following slides. The process of transcription consists of 3 steps: initiation, elongation and termination. There is a single DNA dependent RNA polymerase that catalyzes transcription of all types of RNA. RNA polymerase and the sigma factor bind to the promoter and initiate transcription. It uses nucleoside triphosphatase as substrate and polymerizes in a template dependent fashion following the rule of complementarity. It somehow facilitates opening of the helix and continuous elongation. Only a short stretch of RNA remains bound to the enzyme. Once the polymerase reaches the terminator region, the nascent RNA falls off. So also the RNA polymerase. This results in termination of transcription. The termination of transcription takes place with the help of a factor called Rho factor. See, next I am adding an animation to understand the transcription process very easily. Thanks to DNA Learning Center for providing the animation. In this 3D animation, the pink colored double-stranded DNA in which the initiation factors like RNA polymerase and sigma factor bind to the DNA strand, which start to unwind the double-stranded DNA, and 2 strands will form. One is called the template strand and the other is the coding strand. From the template strand, RNA is produced, that is coming through the groove, that is yellow in color, which elongate. Continuously one RNA strand is produced. The yellow colored substances are substances required for the synthesis of RNA, which moves, and finally the RNA polymerase and mRNA fall off when termination factor called Rho factor bind to this RNA polymerase, this RNA strand and RNA polymerase will fall off and transcription is completed. Next, we can see transcription in eukaryotes. Unlike in bacteria, the eukaryotic genes are called split genes. Why it is called a split gene? Or the DNA contain coding and non-coding region. The coding regions are called exons and the non-coding regions are introns. So this is some bit complex than prokaryotic transcription. Here there are many processes take place like splicing, capping and polyetailing process are there. So we can see through the following slide how the eukaryotic transcription take place and what are the major complexities in eukaryotic transcription. In eukaryotic transcription, the RNA having clear-cut division of labor. The RNA polymerase 1 transcribes rRNAs like 28S, 18S and 5.8S, whereas RNA polymerase 3 is responsible for transcription of tRNA, 5srRNA and snRNAs. The RNA polymerase 2 transcribe precursor of mRNA, that means heterogeneous nuclear RNA, hnRNA. After this, another complexity is splicing, capping and tailing. We can see what are these terms represent. Capping. At the end of transcription, the 5' end of the RNA transcript contains a free triphosphate group since it was first incorporated nucleotide in the chain. The capping process replaces the triphosphate group with another structure called the cap. The cap is added by the enzyme guanyl transferase. Splicing. The process of removal of introns and exons are joined together. There are two main steps in splicing. In the first step, the pre-mRNA is cut at the 5' splice site, the junction of the 5' exon and the intron. The 5' end of the intron then is joined to the branch point within the intron. This generates a lariat-shaped molecule characteristic of the splicing process. In the second step, the 3' splice site is cut and the two exons are joined together and the intron is released. Polyetailing. Polyadenylation is the addition of polya tail to a messenger RNA. The polya tail consists of multiple adenosine monophosphates. In other words, it's a stretch of RNA that has only adenine bases. The tail is shortened over time and when it is short enough, the mRNA is enzymatically degraded. Dear viewers, from these descriptions and explanation, I think you could understand what is transcription and how it is taking place. See you later with another topic soon. So for this, I am adding the upcoming topics in the last slide. Thank you.